I want you to just sit back for a moment. Just rest your hands on your desk and take a good look at them. Look at your fingers.
Yeah, it's an interesting exercise.
Right. Because no matter how perfectly still you try to hold them, there is this biological reality operating just beneath your perception, like a physiological tremor.
Absolutely. You can't escape it.
Now, I want you to imagine you're sitting at a workbench, right? And in front of you is a needle. And you are trying to thread this needle while wearing, like, thick, heavy, cumbersome winter gloves.
Oh, that sounds, impossible.
But to make the physics even more unforgiving, the eye of that needle is completely invisible to your naked eye. And the thread that you're trying to push through that invisible opening, it's moving on its own. It actually has its own motor, its own propulsion system.
That is exactly the environment.
And this is the fundamental, really staggering reality of the clinical embryologist. They are tasked with safely interacting with a single human cell, which we are going to refer to today as an oocyte. And that oocyte measures barely 120 microns across. And they have to introduce a single sperm cell, which is less than 5 microns wide, directly into the architecture of that oocyte.
It's mind-boggling when you really scale it down.
So our mission today on this deep dive is to construct a vivid, mathematically precise mental map of the specialized hardware in the laboratory, the mechanical, optical, and laser systems. We're exploring the architecture of the invisible.
I love that phrasing because it really is the absolute definition of a submicron arena. You know, when we discuss human dexterity, the cultural archetype is usually like the neurosurgeon.
Right. Or a watchmaker, someone with incredibly steady hands.
Exactly. But at the microscopic scale we are discussing today, human biology inherently betrays us. The human nervous system just doesn't fire in a continuous, perfectly smooth stream of data.
It's pulsed, right?
Right. Motor units in your muscles contract in discrete pulses. Your heartbeat creates this systemic volumetric expansion in your blood vessels. Even just the rhythm of your breathing subtly shifts your center of gravity. Wow.
And you cannot train this away through sheer force of will. It's the fundamental baseline of mammalian physiology. So at 400 times magnification, your resting pulse translates into a devastating seismic event.
A seismic event. That's a terrifying way to put it.
But it's true. Therefore, direct manipulation of an oocyte is physically impossible. So the equipment we're dissecting today isn't just like an observational aid. It functions as an absolute physical buffer.
Okay, so if we are dealing with a required physical buffer, that completely shifts my mental model of the laboratory workbench. Like, we shouldn't be picturing a scientist hunched over a desk holding tweezers.
Oh, definitely not.
If you consider the sheer hostility of that microscopic environment to our clumsy macro-scale movements, it sounds less like traditional biology and more akin to piloting a deep-sea research submarine.
That analogy actually maps perfectly onto the physical constraints of the discipline. You have to consider the embryologist as the pilot locked inside a dry, mathematically stable pressure capsule.
Right. They are completely isolated from the crushing pressures and, chaotic thermal currents of the ocean outside. To interact with the trench floor, they don't try to reach their physical hands out into the water.
Obvious not.
Instead, they rely on highly geared, fluid-driven robotic manipulators. They use external sensory arrays and monitors to guide those arms, carefully attempting to pluck a fragile biological specimen from a roof hundreds of meters below.
So the machinery absorbs all their macro-level intent and distills it.
Exactly. It distills it down into micro-adjustments. It is the literal translation of human intent into microscopic action.
Okay, but before we start reverse engineering the mechanics of those robotic arms and, the geometry of the glass tools they wield, we really have to establish the biological parameters of the cells themselves.
We do, because the biology dictates the machinery.
Right, and we are going to be precise with our terminology today. So you will hear us use the term oocyte. That is the exact scientific nomenclature for the unfertilized egg cell. And we really want to be highly deliberate about that pronunciation. Oocyte.
It's important to get that right, yeah.
And we also need to map out the developmental stages of these gametes, specifically what clinical laboratories refer to as the M1 and M2 stages.
Right. So to understand the machinery, you must understand the state of the cell when it actually encounters that machinery. An M1 is an immature oocyte. Biologically, it's locked in the first phase of meiosis.
Meaning it's not ready yet.
Exactly. It hasn't sorted or halved its genetic material yet, so it is biologically incapable of being fertilized.
Hmm.
The machinery can't help an M1. The target phase is the M2 stage.
Okay, M2.
Yes. This is the mature, fully competent stage. During the transition to M2, the oocyte successfully extrudes exactly half of its maternal chromosomes, packaging them into this tiny, tightly bound structure called the polar body.
And that sits on the outside.
It sits just outside the main cellular membrane, exactly. So when an embryologist identifies that polar body, they know the cell is structurally and genetically primed. It is exclusively at this highly delicate M2 stage that the microscopic hardware interfaces with the cell.
Got it. But to interface with an M2 oocyte, you first have to establish visual dominance over the workspace. You have to see the cell and physically reach it at the exact same time.
Which is harder than it sounds.
Right, because if you think about the geometry of like a standard high school or university compound microscope, those two actions are mutually exclusive.
Oh, completely.
You've got the glass slide on a little platform, the light source blasting up from the bottom, and the objective lenses, those heavy metal cylinders with the glass inside there hovering on a rotating turret barely a millimeter above the sample.
Anyone who has used one of those microscopes remembers that inherent anxiety, right? Turning that coarse focus knob, absolutely terrified that you're going to drive that heavy metal lens straight down and crush the glass slide.
Yes, because the focal length requires the glass to be practically touching the sample.
Yeah.
And if you have a massive, bulky metal lens block sitting directly on top of the Petri dish containing your oocyte, the airspace is completely occupied. So where do your surgical tools go? How do you introduce a 5-micron needle into that environment? The clearance is 0. You have a visual field, but you've sacrificed all operational access.
Which is precisely why clinical micromanipulation required a fundamental redesign of optical physics. They had to completely abandon the traditional upright model and adopt what is known as the inverted microscope design.
Now, the term inverted implies a simple flip, but structurally, we're talking about redesigning the entire optical axis, aren't we?
Oh, it's a massive shift. We are dissecting the microscope and reversing its vertical orientation entirely. In the inverted architecture, the light source, the condenser, the phase contrast, rings, all the things that deliver and shape the light, those are mounted high above the workbench, shining downward onto the sample.
Okay, so the light is on top.
Right. And the critical components, the objective lenses that actually perform the magnification and gather the image, those are housed deep underneath the stage. Looking upward, yes. They are reading the visual data through the bottom of the specialized optical plastic Petri dish where the cells are resting.
Now, I want to push back on the necessity of this.
Yeah.
Because engineering in an inverted optical path introduces massive complications, right? There's suddenly forcing the lens to look through the thick plastic bottom of a Petri dish rather than a paper-thin glass cover slip.
You are.
Which means you have to redesign the refractive index correction of the lenses themselves. So wouldn't it have been simpler to just engineer longer objective lenses with a higher working distance? Or, I don't know, design surgical needles that approach the cell at a really steep vertical angle so they could slip past a standard overhead lens?
It definitely seems like a simpler mechanical fix, but doing so violates the strict laws of optics and fluid dynamics. Let's address the optical constraints first. The resolving power of a lens, its ability to distinguish two separate points, like the head and the tail of a 5-micron sperm cell, is governed by its numerical aperture.
Numerical aperture.
Right. To achieve a high numerical aperture and high magnification, the lens must capture as wide an angle of light scattering off the specimen as possible. And physics dictates that to capture that wide angle, the front element of the lens must be physically intimate with the subject.
It has to be close.
It has to be extremely close. If you try to build a lens with a working distance of, say, 2 inches just to allow room for tools, your numerical aperture plummets. You lose the resolution required to actually see the gametes. You simply cannot cheat Abbe's diffraction limit.
So the lens has to be close, regardless of where you put it, which means if it's on top, it acts as the physical ceiling.
Precisely. And regarding your second point, why not just angle the needles steeply down from the top?
Yeah, why not?
Because you are working in a fluid medium. The cells are submerged in a drop of culture media covered by a layer of mineral oil to prevent evaporation. If you introduce a glass microtool at a sharp, say, 80-degree downward angle, you are intersecting the surface tension of that fluid system at a highly unstable vector.
Oh, I see.
The needle basically acts like a wedge. It distorts the optical field with a massive meniscus, and you lose all fine horizontal control over the gametes. So the inverted axis is not a luxury. It's mathematically mandatory.
That makes perfect sense.
By moving the heavy optics beneath the stage, you evacuate the air space above the cell. You unlock this massive, entirely unobstructed hemisphere of working space. And that spatial freedom allows the embryologist to introduce their tools horizontally, sliding in parallel to the floor of the dish to achieve perfect geometric and fluidic control.
Wow. We've essentially stripped the ceiling off the surgical theater.
We have.
And this architectural change completely dictates the human element, too. Because if you were to walk into an IVF lab, you wouldn't see scientists actively moving around the bench, staring through an inverted setup while manipulating horizontal tools. It forces the embryologist into a highly specific, static, ergonomic posture.
It creates a strict biomechanical lock. The embryologist has to sit perfectly still with their eyes fixed into the binocular eyepieces. Their forearms must rest heavily and securely on the workbench to provide absolute grounding for their skeletal structure.
So no floating elbows?
None. Only their fingertips are permitted to make micro-adjustments to the dials and joysticks. They are anchored into this posture for hours at a time, managing their own respiration and posture to ensure their body basically becomes a stabilized extension of the microscope frame.
That's intense. But opening up that airspace above the Petri dish, it exposes the cell, right?
Yes.
Having the physical room to maneuver a needle is only relevant if the cell survives long enough to be manipulated.
Exactly.
And if the environment within that open space isn't completely insulated from the chaotic, wildly fluctuating parameters of the laboratory room, the M2 oocyte is going to undergo catastrophic degradation.
It will.
So we have to analyze the environmental shields, the thermal foundations, and the tremor isolation protocols.
Right. And this is where the discipline pivots from optical physics to environmental thermodynamics. Human gametes, particularly the M2 oocyte, possess zero internal capacity for thermoregulation.
Because normally they're inside the human body.
Exactly, where they are shielded by massive thermal mass. Once extracted, they must operate in an artificial space completely devoid of temperature fluctuations, and the biological vulnerability is primarily located in a microscopic internal scaffolding called the meiotic spindle.
Right. You mentioned earlier that the M2 stage is defined by its readiness to accept genetic material. So the spindle is the structure managing that genetic material.
Yes. The meiotic spindle is constructed from delicate protein polymers called tubulin. These fibers hold the cell's chromosomes in a highly tensioned, perfect equatorial alignment, waiting for the introduction of the sperm's DNA to complete the meiotic division.
Okay.
And these tubulin polymers are deeply, fundamentally reliant on a specific thermal kinetic energy to maintain their structural integrity.
So if the ambient temperature drops, the kinetic energy of the system drops, and the proteins essentially lose their grip.
We call it cold shock depolymerization. If the temperature of the culture media surrounding that oocyte drops from the human physiological norm of 37.0 degrees Celsius down to even, say, 35 or 34 degrees, the tubulin fibers begin to literally unravel.
They go apart.
The structural scaffolding collapses within minutes, and if the spindle falls apart, the chromosomes scatter haphazardly throughout the cytoplasm. So even if you were to successfully inject a sperm cell after that point, the genetic integrity of the cell is irreversibly compromised. The resulting embryo would be profoundly aneuploid, meaning it possesses an incorrect number of chromosomes.
Which presents a massive engineering challenge for the inverted microscope.
Yeah.
Because a standard microscope stage is typically just a thick slab of steel, aluminum, or standard glass. And in thermodynamics, a thick slab of ambient temperature metal is basically a giant heat sink. The moment you place a thin plastic petri dish onto that stage, the metal will aggressively conduct the heat away from the culture media, plunging the oocyte into cold shock.
It sucks the heat right out.
And you can't just aim a space heater at the microscope. That would create turbulent air currents and completely uneven heating.
Right, you require highly localized, perfectly uniform, and entirely transparent heat. So the solution borrowed heavily from aerospace and advanced optoelectronics. Indium tin oxide, or ITO.
Indium tin oxide.
Yes. The embryologist doesn't place the petri dish on cold metal. The inverted microscope features specialized, optically clear glass insert plates that sit directly above the objective lenses.
Wait, but standard glass is an insulator, not a heating element.
Which is why these specific glass plates undergo a highly specialized vacuum deposition process. They are coated with a microscopic, atomically thin layer of indium tin oxide. ITO is an incredibly unique material. It's a wide bandgap semiconductor.
Okay, what does that mean in practical terms?
What that means is its atomic structure allows photons in the visible light spectrum to pass right through it unimpeded. So it has perfect optical clarity. You can look right through it. However, it retains the properties of a metal when it comes to electrical conductivity.
Wow. So you have engineered a metal layer so thin that it acts like a window, but you can still run a current through it.
Exactly. The workstation's environmental control unit runs a continuous, meticulously regulated low-voltage electrical current across that clear microscopic metal layer. As the electrons move through the ITO matrix, they encounter electrical resistance, which generates heat.
That's brilliant.
This transforms the entire clear viewing window into a highly uniform automated thermal plate. It gently radiates heat upward through the plastic petri dish, maintaining the internal microdrop of culture media holding the oocyte at exactly 37.0 degrees Celsius.
Let's discuss the tolerances of that system, because maintaining exactly 37.0 degrees in a room where the ambient air conditioning might be fluctuating between like 20 and 22 degrees, that requires a serious feedback loop.
The clinical tolerance is typically plus or minus 0.1 degrees Celsius.
0.1. That is an astonishingly tight margin.
It is. To achieve this, the ITO glass isn't just a dumb heater. It is embedded with highly sensitive microscopic thermistors. These sensors are constantly reading the surface temperature multiple times a second and feeding that data back to a proportional integral derivative, or PID, controller.
Okay.
So if a draft of cold air from the laboratory HVAC system sweeps across the stage, the thermistors detect the microscopic drop in thermal kinetic energy. The PID controller instantly calculates the required offset and marginally increases the electrical voltage to the ITO layer, countering the cooling effect in real time. It acts as an active computational thermal fortress.
Man. So we have mathematically solved the thermodynamics. The oocyte is locked at 37.0 degrees, and the tubulin polymers of the meiotic spindle remain tightly bound. But thermodynamics is only one energy vector acting on the workspace. We have to address the kinetic energy of the environment, the sheer physical stability of the room itself. You mentioned the physiological tremor of the human operator earlier, but the building hosting the laboratory presents its own seismic profile.
It does, and this is where the physics of extreme optical magnification profoundly distort your perception of physical reality. Because to a human being standing in a room, a modern building feels entirely solid. It feels like a static object.
Right.
But an IVF laboratory is typically embedded within a larger hospital complex or a multi-story commercial structure.
And those structures are incredibly noisy from a mechanical standpoint. You have massive air handling units on the roof spinning heavy turbines, elevator cars dropping down shafts on steel cables.
You have heady fire doors slamming shut 3 corridors away, the deep, low-frequency rumble of city buses passing on the street outside, or even just the rhythmic impact of a colleague's footsteps walking across the laboratory floor. Because of your own mass and the dampening of your nervous system, you don't consciously perceive these vibrations. But vibration is merely kinetic energy traveling as a waveform through a solid medium. When those waveforms travel up the legs of a standard desk through the microscope frame and hit the specimen stage, you are suddenly looking at them under 400x magnification.
It's magnified too.
Exactly. At that scale, a footstep looks like a violent, rolling earthquake tearing through your field of view. The cell literally bounces out of focus.
The scale of disaster here is terrifying.
Yeah.
If an embryologist is in the middle of a procedure, right, and they have successfully breached the membrane and inserted a microscopic glass needle into the cytoplasm of the oocyte, and a heavy delivery truck hits a pothole outside.
It's a nightmare scenario.
That low-frequency kinetic waveform travels through the bedrock, up the building's steel skeleton, through the floor, and into the microscope. If that glass needle vibrates laterally, even by 2 or 3 microns, while inside the cell,
It acts like a microscopic windshield wiper. The lateral kinetic shear force will instantly tear the internal cytoskeleton, rupture the intracellular organelles, and completely liquefy the oocyte. The procedure is over.
So how do you defeat the kinetic energy of a multi-ton building?
Yeah.
I mean, you can't just bolt the microscope to the floor,
Right.
because the floor is the source of the waves.
Right.
You have to somehow disconnect the microscope from the architecture of the building.
You achieve isolation through pneumatic levitation.
Levitation.
Yes. The inverted microscopes don't sit on standard laboratory casework. They are stationed on highly specialized pneumatic anti-vibration tables. These tables are engineered around a massive, heavily damped tabletop, often made of thick, stainless steel layered over a dense honeycomb core to provide massive structural inertia.
Okay.
But that dense tabletop is not physically bolted to the legs of the desk.
It's floating.
It is entirely suspended. Underneath the four corners of that heavy tabletop are pneumatic isolators. They are highly pressurized bladders or cylinders filled with compressed air. The entire optical and mechanical weight of the microscope system is essentially floating on these active cushions of gas.
Wow. This makes me think of, like, high-end active noise-canceling headphones. But instead of trying to read and invert acoustic sound waves in the air, this pneumatic system is passively absorbing and canceling the physical low-frequency seismology of the building structure itself.
That's a great analogy. The fluid dynamics of the compressed air act as a low-pass mechanical filter. When the kinetic wave from that delivery truck travels up the metal leg of the table, it hits the compressed air bladder. Because gas is highly compressible, the wave expends its energy slightly compressing the air inside the bladder rather than transferring the hard kinetic shock into the steel tabletop above.
That is so smart.
Furthermore, many of these systems are self-leveling. They contain mechanical valves linked to an external pressurized air supply. If the embryologist leans their weight slightly onto the front edge of the table, the valves instantly detect the shift in load and pump more air into the front bladders to keep the optical plane perfectly level to within a fraction of a millimeter.
Incredible.
It renders the massive kinetic chaos of the building mathematically silent.
Okay, so the environment is now strictly controlled. We have an inverted visual field carving out the physical airspace above the cell. We have a transparent semiconductor stage actively calculating and maintaining a thermal baseline of exactly 37.0 degrees.
Yeah.
And we have a high-mass table floating on compressed nitrogen or air to sever the workstation from the building's gravity and vibration.
Yes.
The stage is perfectly warm, and it is perfectly mathematically still.
Yeah.
It is only at this point, after layering all these passive defensive shields, that the embryologist can finally introduce motion back into the system.
Exactly. Now we can move.
Which brings us to the mechanical bridge, the interface that safely translates the embryologist's intent into submicron action, the micromanipulators.
We can conceptualize this as the joystick bridge. If you look at the architecture of the workstation, bolted directly to the heavy, vibration-isolated frame of the microscope are two intricate mechanical towers.
One on each side.
Yes. Extending down from these towers, positioned ergonomically near the embryologist's resting hands, are two joystick controllers. These are the physical translators. They absorb the macroscopic, inherently flawed movements of a human hand and mathematically gear them down into flawless submicron needle movements at the cellular level.
To understand the power of that translation, we really need to quantify the scaling factor. If I am holding that joystick and I make a deliberate movement, what is the mathematical ratio between the distance my hand travels and the distance the needle travels under the lens?
The scaling factor is staggering, and it's often adjustable depending on the phase of the procedure. These manipulators use proportional translation ratios typically configured around 100 to 1, or for extremely fine work, up to 500 to 1.
So if I move the joystick.
If you push the joystick forward by a large, easily controllable distance of 5 millimeters, a physical sweep of your hand that you can easily track with your own eyes, the mechanical bridge severely restricts that input. The needle under the lens moves merely 10 to 50 microns.
That's amazing.
The system takes the clumsy, pulsing, vibrating energy of your human arm and distills it down into pure, mathematically smooth geometric progression.
I really want to break down exactly how that gearing is achieved physically, because there are two distinct schools of engineering used in modern labs to build this bridge: analog oil hydraulics and digital electronic steppers.
Right.
Let's start with the analog system, because the idea of using mineral oil to perform submicron cell surgery seems almost primitive until you understand the fluid dynamics behind it.
It does sound primitive, but the oil hydraulic micromanipulator is truly a masterpiece of pure analog engineering. It bypasses electronics entirely in favor of fluid mechanics. It operates on a closed-loop, master-slave piston cylinder architecture.
Okay, master-slave.
Right. At the base, inside the joystick housing, you have the master piston. Up on the microscope tower, physically holding the micro tool, you have the slave piston. Linking these two cylinders are thick, heavily shielded, flexible Teflon tubes.
And inside the tubes,
The entire internal volume of this closed system is filled to absolute capacity with a medical-grade, meticulously degassed mineral oil.
Okay, but if the goal is absolute precision, why rely on a fluid? Why not just use highly machined metal gears, racks, and pinions to step the movement down?
Because metal gears suffer from the physical reality of machining tolerances. If you build a gearbox, no matter how exquisitely you polish the metal teeth, there must always exist a microscopic gap between those teeth so they can slide past one another without seizing.
Right, they need a little room to turn.
Exactly. And that gap creates a phenomenon called mechanical backlash.
Ah. Like, if you are turning a gear clockwise, and you suddenly want to reverse direction and turn counterclockwise, there's a tiny fraction of a second, a tiny rotational degree, where the first gear has to close that gap and slam into the back of the second gear before the movement catches.
Yes. At the macroscopic level of driving a car, you barely feel it. But under 400x magnification, navigating inside a 120-micron cell, a tiny jump is a violent, catastrophic lurch. It will tear the plasma membrane wide open.
Wow.
So the oil hydraulic system completely eliminates mechanical backlash by eliminating the gears. Because mineral oil is a liquid, its bulk modulus is incredibly high. For the practical purposes of this machinery, it is functionally incompressible.
Right.
When you gently tilt the joystick, you are driving the master piston forward, displacing a highly specific microscopic volume of oil. Because that oil cannot compress, that exact volumetric displacement must travel instantly up the Teflon tube and push the slave piston holding the needle by the exact same proportional volume.
And because it is a continuous column of fluid, there are no gaps. There is no slack to take up when you reverse direction.
None. The fluid dynamics provide a continuous, highly dampened, perfectly linear transfer of force. It feels completely organic.
But there's a thermodynamic vulnerability here, right? We just spent 20 minutes discussing how tightly the lab controls temperature. If the ambient temperature of the ream fluctuates, physics dictates that the mineral oil inside those tubes will undergo thermal expansion or contraction.
That's true.
So if the oil expands, wouldn't that push the slave piston and cause the needle to drift forward on its own, completely ruining the calibration?
That is an incredibly sharp deduction, and it was a major historical hurdle in the early days of micromanipulation. Thermal drift is the enemy of analog hydraulics.
So how do they fix it?
Modern systems combat this in a few ways. First, the total volume of oil in the system is kept as low as possible to minimize the gross volumetric expansion. Second, the Teflon tubing is heavily thermally shielded. And third, the labs rely on the massive thermal stability of the room's HVAC system to keep ambient fluctuations to an absolute minimum.
Okay, that makes sense.
However, because of this vulnerability, many laboratories have transitioned to the second school of engineering, electronic micromanipulators.
Let's talk about those. How does the architecture change when we move from analog oil to digital electronics? We're removing the fluid completely, right?
We remove the fluid entirely and replace it with high-resolution stepper motors, piezoelectric drives, and digital optical encoders. The joystick is no longer pushing a physical master piston. It is essentially a fly-by-wire system.
Like a modern jet.
Exactly. The joystick sends purely digital encoded electronic signals to a microprocessor. The processor calculates the required translation ratio and sends electrical pulses to the tiny stepper motors mounted on the towers, instructing them to advance or retract the needle in discrete, mathematically perfect increments.
So you lose the vulnerability of thermal expansion and you gain computational control. You could, program the microprocessor to remember specific coordinates in three-dimensional space, allowing the motor to withdraw the needle, grab another sperm cell, and automatically return the needle to the exact same focal plane with a single button press.
Precisely. Electronic systems are incredibly advantageous for high-throughput setting or heavily automated clinical workflows. They never suffer from fluid drift.
But I imagine the human experience of operating these two systems is vastly different. If you're an embryologist spending 8 hours a day anchored to this machine, do you develop a different somatic feedback loop, a different muscle memory, depending on whether you are pushing analog oil or sending digital signals?
Oh, absolutely. It alters the entire sensory experience of the procedure. Veteran embryologists often speak about the oil hydraulic systems with a deep sense of tactile reverence.
Tactile reference, I like that.
It's true. Because you are physically pushing a closed column of dense fluid, you actually feel a microscopic dampened resistance pressing back through the joystick. You develop a deep, intuitive muscle memory for the viscosity of the oil. It feels grounded. It feels like an organic extension of your own nervous system.
You can feel the physics of the movement.
Yes.
Mm.
With the electronic manipulators, that tactile feedback loop is severed. You are providing directional input to a computer sensor. The joystick feels feather-light, completely detached from the physical resistance of the motor towers.
Right.
It is mathematically pristine, but somatically sterile. It's the difference between flying a vintage biplane with physical cables connecting the stick to the ailerons versus flying a modern fighter jet where your stick merely sends electrical commands to hydraulic pumps. Both achieve the objective, but the physical dialogue between the human and the machine is fundamentally altered.
We have built an incredible mechanical superstructure here. We have the thermal isolation, the kinetic isolation, the inverted optics, and now the joystick bridge, scaling our macroscopic intent down to sub-micron precision. We have the shoulder and the arm of the operation.
I do.
But we have to talk about the fingers. What are the actual physical objects intersecting with the biological membrane of the cell? We obviously aren't using microscopic metal tweezers.
Metal is completely unviable at this scale. Even the most highly polished surgical steel possesses an inherently rough, jagged, crystalline structure when viewed at 400x magnification.
It would just tear everything up.
It would act like a rasp against the delicate lipid bilayer of the oocyte. Furthermore, metal is opaque, which would block the optical path.
Good point.
The tools that physically interact with the gametes are fabricated exclusively from what we call architectural glass. These are custom manufactured micropipettes, pulled from high-purity borosilicate glass capillaries down to breathtakingly tight geometric tolerances.
Let's map out the specific geometry of these glass tools.
Yeah.
In a standard clinical procedure like intracytoplasmic sperm injection, which the field refers to as ICSI, the embryologist is operating two distinct tools simultaneously, one controlled by the left joystick, one by the right. Let's analyze the left hand first, the tool responsible for securing the cell.
Right. The tool in the left manipulator tower is called the holding pipette. Its singular structural purpose is to capture the M2 oocyte, stabilize it against the chaotic fluid dynamics of the culture drop, and keep it perfectly stationary.
Okay.
If you examine the holding pipette under the inverted microscope, you will see a relatively thick outer diameter, often around 100 to 120 microns, which is roughly the same size as the oocyte itself. This provides massive structural rigidity.
But the tip cannot be sharp. If you press a sharp 100-micron glass tube against the cell, you will crush it.
Which is why the manufacturing process of the holding pipette involves a technique called microforged fire polishing. The very tip of the glass capillary, the opening that physically contacts the cell, is exposed to a microscopic, highly controlled, heated platinum filament.
Oh, wow.
This briefly melts the leading edge of the glass, causing surface tension to pull the rough edges inward. It creates a completely flat, perfectly rounded, donut-shaped opening.
It essentially functions like a microscopic polished vacuum hose.
That is the exact mechanical principle. The back end of this holding pipette is connected through airtight lines to a highly sensitive microsyringe filled with oil or air. The embryologist maneuvers the flat, polished face of the holding pipette directly up against the outer shell of the oocyte. They then turn a finely threaded dial on the syringe to generate a very gentle, meticulously controlled negative pressure.
Suction.
But you have to balance that suction perfectly. If you apply too much negative pressure, you don't just hold the cell. You will literally vacuum the intercellular mass, the cytoplasm, right out of the shell and into the glass tube, destroying it. You are balancing the pressure gradient against the structural elasticity of the cell's outer shell. When done correctly, the flat, polished opening gently grips the curvature of the oocyte, holding it rigidly in place without drawing it inside. The holding pipette acts as the stationary anvil, and once the cell is locked against the anvil, you can bring in the hammer.
The hammer is loaded in the right hand.
Yeah.
The ICSI injection needle. This is where the geometry transitions from stabilization to active penetration.
The ICSI injection needle is the offensive micro tool. Its operational mandate is to capture a single selected sperm cell, carry it through the fluid, penetrate the tough outer shell of the oocyte, pierce the elastic inner membrane, and deliver the payload.
Yeah.
The physical dimensions of this needle are terrifyingly precise. It is a hollow glass tube, but it is drawn out until the outer diameter of the tip is merely 5 to 7 microns.
Let's contextualize that scale for a second.
Yeah.
A standard red blood cell is about 8 microns wide. A single strand of human hair is roughly 70 microns thick. This hollow glass needle is less than 1/10 the thickness of a human hair, and it is entirely hollow, so fluid and a sperm cell can pass through the center.
Yes. But a simple flat tube cannot penetrate the membrane. The tip of the injection needle is aggressively beveled, cut at a sharp, highly specific angle to create a cutting edge. And at the very apex of that 5-micron bevel, the manufacturers often engineer a microscopic internal spike, or a tiny, inward-pointing glass tooth.
What is the mechanical purpose of that internal tooth? Why not just use a smooth, sharp point like a hypodermic needle?
Because the inner membrane of the oocyte, the ulema, is incredibly elastic. It behaves less like a rigid wall and more like a thick latex balloon. If you simply press a smooth, sharp needle against it, the membrane will often stretch and deform deeply inward, wrapping around the needle without actually breaking.
Just denting it.
Right. You end up pushing the membrane against the far wall of the cell, severely stressing the internal cytoskeleton. That tiny internal glass tooth acts as a snag point. As the beveled edge stretches the membrane, the microscopic spike catches the lipid bilayer, creating a localized point of massive mechanical stress that forces the membrane to cleanly snap and rupture over the tip, allowing the needle to slide inside with minimal geometric distortion of the cell.
That is an astonishing application of microscopic mechanical friction. But there's another geometric detail about these glass needles that challenges your intuition. If you look at them straight out of the sterile packaging, the injection needles are not perfectly straight tubes. They are permanently pre-bent near the tip.
Yes, they are.
If you want absolute linear control, why would you introduce a bend into your primary surgical tool?
Because the manufacturing must compensate for the architectural limitations of the workstation itself. Think back to our discussion on spatial freedom. The objective lens is underneath, looking up. The holding pipette and the injection needle are mounted to the manipulator towers, which sit on the left and right sides of the microscope frame.
Right.
To reach the cell resting in the center of the heated stage, the tool holders must angle slightly downward, approaching the fluid drop from a superior oblique angle.
So the robotic arm is swooping down toward the dish, maybe like a 30-degree angle, relative to the flat floor of the Petri dish.
Correct. If you inserted a perfectly straight, rigid glass needle into that angled tool holder, the tip of the needle would plunge into the culture drop and strike the floor of the Petri dish at that exact same 30-degree downward angle. It would point directly into the plastic.
And you wouldn't be able to lay the needle flat along the floor to scoop up a sperm cell.
Yeah.
You'd just be digging into the plastic substrate.
Exactly. You would lose all horizontal operational capability. So during the advanced manufacturing process using precise microforges, the glass capillaries are heated and permanently pre-bent at highly specific angles, typically 30 or 35 degrees.
Oh.
The angle of the bend is calculated to perfectly invert the approach vector of the micromanipulator arm. By the time the beveled tip of the glass needle reaches the microscopic field of view under the objective lens, the pre-bend has perfectly leveled it out.
That's genius.
The final few millimeters of the glass are running absolutely parallel to the flat floor of the heated stage, allowing the embryologist to sweep the needle smoothly across the bottom of the dish to capture the sperm.
It's brilliant physical precompensation. Okay, so we've established the geometry of the glass, but as we noted, these are not solid glass pokers. They're hollow capillaries. Their entire function relies on moving biological material through their hollow core,
Yes.
which requires us to analyze the fluid dynamics of the system. How does an embryologist generate and control the microscopic flow of liquid inside a 5-micron tube?
This is the domain of picoliter fluidics, and managing this physical pressure gradient is arguably the most demanding tactile skill an embryologist must master. They utilize highly geared micro-injector pumps. These are typically manual micrometer threaded syringes, heavily geared to translate large rotational turns of a dial into microscopic linear displacements of a plunger, which pushes or pulls air or oil through the lines connected to the back of the injection needle.
When we talk about displacing fluid at this scale, we use the term picoliter. Let's frame that measurement in relation to the physics of the cell. If a milliliter is a small thimble of fluid, and a microliter is a tiny visible drop on the end of a pipette, a picoliter is 1/1,000,000th of a microliter.
It's unfathomably small.
When you're dealing with picoliter volumes, standard macroscopic fluid dynamics begin to break down. You aren't just moving liquid, you are actively fighting the surface tension and capillary action of the fluid on a microscopic scale.
You are fighting the inherent physical properties of the water molecules themselves. By turning the finely threaded dial on the microinjector pump, the embryologist creates a microscopic negative pressure, a vacuum, at the very tip of that 5-micron needle. They carefully hover the needle over a single, highly selected sperm cell swimming in the viscous media and use that vacuum to draw the sperm up into the shaft of the glass.
And there's a specific physical protocol for how that sperm must be drawn into the needle.
Uh-huh.
They don't just vacuum it up randomly. They intentionally draw it in tail first.
The orientation is critically important for two reasons. First, the head of the sperm contains the absolute payload, the tightly packed chromatin containing the male genetic material. When the embryologist pushes the fluid back out to deliver the sperm into the oocyte, you want the heavy head of the sperm to be the first biological structure to exit the needle and enter the cytoplasm. Secondly, the physical act of drawing the long, rapidly whipping flagellum, the tail, backward across the sharp internal glass edge of the needle bevel is often used as a deliberate mechanical technique. It physically damages the flagellar membrane, permanently immobilizing the sperm.
Because if you inject a highly motile swimming sperm directly into the core of the oocyte, it will literally thrash around inside the cytoplasm, physically turning up the organelles and destroying the cell from the inside out.
Exactly. You must neutralize its kinetic energy before introduction. Once the immobilized sperm is loaded in the needle, the embryologist moves across the petri dish, aligns the needle with the holding pipette, and pierces the M2 oocyte.
This is the moment.
This is where the mastery of picoliter fluid pressure becomes absolute. When the tip of the needle is resting deep inside the inner cytoplasm of the cell, the embryologist must reverse the dial on the micro-injector pump, applying positive pressure to push the fluid and the sperm out.
But they have to manage that pressure imbalance perfectly. If you push the dial too aggressively, you aren't just delivering the sperm. You are injecting a massive, uncontrolled volume of the surrounding chemical culture media directly into the closed environment of the egg.
Which is catastrophic.
You will blow the cell up from the inside like a microscopic water balloon until the membrane just bursts.
Cytoplasmic blowout. It is a fatal error. Conversely, after the sperm is deposited, the embryologist must slowly withdraw the glass needle from the cell. If there is even a fraction of negative suction remaining on the line, if the fluid dynamics are not perfectly neutral, the retracting needle will act like a vacuum, sucking the delicate inner cytoplasm and potentially the meiotic spindle right out of the cell, irrevocably destroying it upon exit.
Wow.
The fluid mechanics must be balanced to an absolute zero pressure state at the exact millisecond of withdrawal.
That level of somatic control over fluid physics is awe-inspiring. We have built an incredible mechanical system relying on the sharp physical geometry of glass to breach the cell. But there are clinical scenarios where utilizing blunt mechanical force, even with a perfectly engineered 5-micron beveled glass spike, is simply too risky or inefficient.
That's true.
If the outer shell, the zona pellucida, is unusually hardened, attempting to force a needle through it threatens to physically crush the entire cell against the holding pipette. When the laboratory needs to bypass mechanical friction entirely, they switch from utilizing architectural glass to utilizing the physics of pure light. This brings us to the domain of laser mechanics.
This represents one of the most significant technological leaps in the history of clinical embryology. To breach the protective outer shell of the embryo, whether to assist the embryo in hatching out of its shell, or to create an opening to safely extract a few cells for genetic testing, we no longer rely on the archaic methods of applying acidic chemical tyrrhode solution or creating mechanical friction with glass needles.
So what do we use?
Modern laboratories utilize non-contact, highly targeted infrared lasers that are seamlessly integrated directly into the inverted microscope's primary optical path.
I really want to break down the biophysics of this.
Okay.
Because the moment you tell a patient or a layperson that you are literally firing a medical laser at a developing human embryo, it sounds terrifying.
I know, it does.
It sounds like science fiction bordering on recklessness.
May it.
We need to explain exactly what this laser is doing at the atomic level, because the safety profile relies entirely on thermodynamic timing.
The fear is totally understandable, but the physics are beautifully elegant. Let's look at the target material. The zona pellucida is a thick, highly elastic glycoprotein matrix that completely surrounds the oocyte and the early dividing embryo.
Okay.
Conceptually, think of it as a porous, highly hydrated sponge made of specialized proteins and sugars. The clinical laser integrated into the microscope operates at a highly specific immutable wavelength in the infrared spectrum, specifically 1480 nanometers.
Why that precise number? Out of the entire electromagnetic spectrum, why did optical engineers isolate 1480 nanometers?
Because of the molecular absorption spectra of water, the glycoprotein matrix of the zona pellucida is heavily hydrated. Water molecules are trapped everywhere within its protein lattice. Physics demonstrates that the exact wavelength of 1480 nanometers aligns perfectly with the vibrational overtone of the oxygen-hydrogen bonds in a water molecule.
So it only targets water.
Exactly. When you fire a laser at 1480 millimeter, the biological proteins essentially ignore the light, it passes right through them. But the water molecules aggressively, almost exclusively, absorb those specific photons.
So the light energy is captured by the water. And according to the laws of thermodynamics, when molecules absorb massive amounts of optical energy, they begin to vibrate rapidly. They convert that light into localized thermal energy, heat.
Exactly. The laser instantaneously superheats the highly localized microscopic water molecules trapped in the target area of the shell. This microscopic flash boiling effect physically dissolves the structural protein matrix, vaporizing a perfectly round, clean microscopic hole in the shell without ever physically touching it.
But wait, if you're generating enough heat to flash boil water and dissolve proteins, how does that heat not instantly radiate inward and boil the living, dividing cells residing just a few microns away inside the embryo? Heat diffusion in a liquid medium is incredibly fast.
And that is where the mastery of thermodynamic timing comes into play. The safety relies entirely on the concept of thermal relaxation time. The laser does not fire a continuous beam. It fires in ultra-short, highly precise pulses controlled by a digital microprocessor. A single firing sequence lasts anywhere from just 0.5 to 2.0 milliseconds.
0.002 of a second.
Yes. The laser dumps the optical energy into the water so fast that the localized heat spikes, dissolves the shell, and instantly dissipates outward into the massive heat sink of the surrounding cold culture media before the thermal kinetic wave even has time to physically propagate inward toward the cellular mass.
That is incredible.
The thermodynamics are so tightly constricted by time that the living cells inside experience a net zero thermal energy transfer. Their internal temperature does not rise by a single fraction of a degree.
I have to play devil's advocate here to address another pervasive fear. Even if we have solved the thermal transfer problem, there is a fundamental anxiety about radiation. The myth is that firing any form of high-energy light at an embryo runs the risk of radiation damage, that stray photons might strike the DNA within the inner cells, causing a genetic mutation that will manifest in the resulting child.
We can categorically and firmly debunk that myth by examining the physical difference between ionizing and non-ionizing radiation. Ionizing radiation, which includes X-rays, gamma rays, and deep ultraviolet light, carries photons packed with massive amounts of electron-volt energy. When those high-energy photons strike an atom, they literally rip electrons out of orbit.
Right, that's what causes the damage.
That atomic disruption is what physically severs the sugar-phosphate backbone of a DNA strand, causing genetic mutations.
But the 1480 nanometer infrared laser sits on the complete opposite end of the energy spectrum.
Correct. Infrared light is strictly non-ionizing. The photons at 1480 nanometers possess highly specific lower energy states. They have enough energy to make a water molecule vibrate and generate heat, but they completely lack the physical voltage required to strip an electron from an atom. It is physically impossible for this wavelength of light to alter atomic structures.
That's a huge relief.
Furthermore, because the 1480 millimeter wavelength is so aggressively absorbed by the water in the outer shell, its optical penetration depth is microscopically shallow. The light physically cannot reach the nucleic acids housed deep within the inner cells. There is zero biological or physical mechanism by which this laser could cause a genetic mutation in the DNA. It functions purely as a highly localized microthermal knife.
That is deeply reassuring and, frankly, a fascinating application of optical biophysics. But building this vast mechanical ecosystem raises a critical operational vulnerability. We spent this entire discussion mapping an incredibly complex interrelated system. We have the inverted optical path correcting for refractive indices. We have indium tin oxide transparent semiconductors actively monitoring thermodynamics via PID controllers. We have compressed air isolators actively defeating the low-frequency seismology of the building structure. We have incompressible mineral oil translating macroscopic kinematics into submicron geometry via architectural glass. And we have non-ionizing microsecond infrared lasers riding down the optical pathways.
It is a phenomenal density of high-precision engineering concentrated into a single desk.
Which means the potential for systemic drift is massive. In the physical world, entropy is constant. Mechanical systems wear down, glass shifts, thermal sensors degrade. With all these high-tech components required to operate in perfect simultaneous unison to protect the oocyte, how do you stop a system this complex from slowly, silently drifting out of mathematical alignment?
You are identifying the invisible, unsung hero of the entire clinical laboratory framework. preventive metrology.
Metrology.
Metrology is the rigorous scientific study of measurement and calibration. To guarantee continuous, unbroken safety for these highly vulnerable cells, all of this micromanipulation hardware requires obsessive, strictly scheduled preventative maintenance and calibration routines. This is not administrative background work. This is a vital, frontline clinical safety protocol executed by specialized biomedical engineers who understand the physical tolerances of the machinery.
Let's delve into how that physical degradation actually manifests. Because as you noted, the machinery doesn't usually fail with a catastrophic explosion. It suffers from microscopic, insidious wear and tear. What does that drift look like in the context of the hardware we've discussed?
Let's analyze the oil hydraulic lines. Over thousands of procedural uses, pushing and pulling that master piston, microscopic amounts of wear begin to manifest in the internal rubber O-rings and seals. This degradation might allow a microscopically small air bubble to slip past the seal and enter the closed fluid loop.
And because air is a gas, it is highly compressible, unlike the mineral oil.
Exactly. The moment a compressible gas enters the line, you lose the mathematical purity of the fluid dynamics. The joystick movement suddenly feels slightly spongy. You introduce mechanical backlash back into the system, threatening the precise geometry of the needle thrust.
Makes sense.
Or consider the ITO heated glass stages. Over years of continuously conducting electrical voltage across its microscopic matrix, the metal layer can develop subtle imperfections or microfractures, altering the local electrical resistance. A thermistor might report a perfect 37.0 degrees Celsius at the center of the plate, but the far edge of the glass, where the embryologist is staging a secondary culture dish, might subtly drop to 36.4 degrees.
Which introduces the threat of cold shock depolymerization to the meiotic spindle fibers, completely compromising the genetic structure of the cell without the embryologist ever realizing the temperature had dropped.
Precisely. And then consider the laser path. The infrared laser fires down through the optical column, bouncing off precisely angled dichroic mirrors before passing through the objective lens. Even a minor physical bump to the massive microscope frame, say, someone moving the desk slightly during cleaning, could induce a microscopic alignment drift in those internal mirrors.
Oh.
The embryologist might look through the binocular eyepiece, align the digital targeting reticle perfectly over the zona pellucida, and fire the laser. But because the internal mirror shifted, the actual physical beam of light might strike 2 microns to the left of the crosshairs,
Striking the living cell instead of the protective shell. That is a terrifying prospect.
Therefore, to defend the baseline operational integrity, the lab institutes meticulous scheduled metrology audits. The biomedical engineers don't rely on the internal software or the machine's own sensors, because those sensors themselves might be drifting. They bring in independent, highly calibrated physical standards.
Like what?
To audit the laser, they use specialized calibration beads, tiny, perfectly spherical, inert plastic microbeads of known mathematically exact dimensions. They align these beads under the microscope and fire the laser repeatedly, measuring exactly where the thermal ablation occurs relative to the digital targeting reticle. They verify the optical alignment physically down to the fraction of a micron.
And how do you audit the invisible thermal gradients of the ITO metal heater? You can't just stick a standard mercury thermometer into a microscopic drop of fluid.
No, they utilize independent, highly sensitive digital surface thermometers equipped with ultra-fine wire thermocouple probes. The engineer will physically map a grid across the entire surface area of the glass insert, reading the thermodynamic output millimeter by millimeter. They are independently recertifying the physical reality of the heating element against a traceable national metrology standard, ensuring that the 37.0 degree reading is uniform and absolute across the entire functional workspace.
I always wonder what happens when the institutional safety net fails. Let's say a laboratory is facing an incredibly high volume of clinical cases. The staff is overwhelmed, and management decides to delay the biomedical engineering audit. They skip the laser calibration for a month, or they delay servicing the seals on the oil manipulators. How does a minor microscopic misalignment, a slow drift, actually manifest in the lab's day-to-day clinical reality? What happens to the biology?
As I mentioned, the machinery rarely fails loudly. It drifts silently out of specification. And when the physical environment drifts, that mechanical error translates instantly into physical and thermodynamic stress on the cellular biology.
So the cells feel it first.
The embryologist might notice that they are having a slightly harder time getting the injection needle to snap cleanly through the membrane, perhaps because the approach angle has drifted by 2 degrees, causing more geometric distortion. Or they might not notice anything tactile at all, but the oocytes are silently experiencing a half-degree drop in ambient temperature during the critical 10-minute ICSI procedure. How this manifests clinically is a slow, insidious, deeply frustrating drop in the laboratory's key performance indicators.
The data starts to reflect the mechanical drift.
Exactly. The day-to-day fertilization rates will inexplicably dip by a few percentage points. The critical blastocyst conversion rate, the number of fertilized eggs that successfully divide and grow into strong, viable day 5 embryos, will subtly decline across the board.
And they might not know why.
The clinical staff will start searching for biological reasons, questioning the culture media or the incubator gas lines, when the reality is a creeping mechanical failure at the workstation. The absolute unyielding mathematical precision of the micromanipulation machinery is the silent anchor of the laboratory's clinical success. If the anchor drags, the entire operation drifts into failure.
It all routes back to the concept of the physical buffer that we established at the very beginning of this discussion. If the machinery is not geometrically and thermodynamically perfect, the embryologist's hands, no matter how vastly experienced or highly trained they are suddenly exposed to the chaotic, blunt force variables of the macroscopic room.
Exactly right.
We have successfully constructed an incredible, multidimensional mental map today. We started by dismantling standard optical physics, carving out the essential unobstructed vertical airspace via the inverted microscope axis. We learned how aerospace pneumatic engineering floats massive steel tables on compressed air to absorb and silence the low-frequency seismology of the building structure. We rode the joystick bridge, tracking how incompressible medical-grade mineral oil absorbs a clumsy, sweeping human hand motion and scales it down into a flawless, mathematically continuous, microscopic crawl.
It's quite a journey.
We explored the exquisite geometry of architectural glass pipettes, from the fire-polished flat vacuum of the holding needle to the 5-micron beveled spike designed to snag an elastic membrane. We analyzed picoliter fluid mechanics, neutralizing pressure gradients to avoid cytoplasmic blowout. We fired non-contact infrared lasers tuned to 1480 nanometers, dissolving protein shells with microsecond bursts of thermodynamic energy while leaving the inner DNA perfectly cold and completely untouched. And finally, we grounded this entire fragile ecosystem in the rigorous, obsessive metrology audits that keep the mechanical symphony in perfect tune.
If we attempt to connect all of these highly disparate engineering disciplines into a final, unified framework, I believe there is a profound, almost philosophical realization to be found.
Oh, I'd love to hear it.
When the general public, or even patients, hear about the integration of microsecond infrared lasers, robotic pneumatic tables, and automated semiconductor heating elements, there is a natural human tendency to feel alienated. It can feel as though the deeply biological process of early cell development is becoming overly sterile, dangerously industrialized, or removed from nature.
Right, sounds very cold.
But the reality is the exact opposite. This staggering array of advanced machinery does not distance the laboratory from the natural needs of the biology. Instead, it serves as what we might conceptualize as a technological shield. It represents the highest ultimate expression of manual clinical safety. The laboratory is deploying the bleeding edge of optical physics, fluid dynamics, and thermodynamics to build an absolute impregnable physical wall. A wall meticulously designed to isolate the fragile, highly sensitive potential of early biological life from the chaotic, wildly fluctuating, and inherently hostile variables of the macroscopic human environment. The heavy machinery exists exclusively to protect and serve the microscopic biology.
A technological shield. That is an incredibly beautiful and highly provocative thought to carry forward. The idea that all of this heavy steel, cold architectural glass, and tightly regulated electrical voltage is fundamentally designed to wrap its mechanical arms around something inconceivably delicate, protecting it from our own inherent physical clumsiness. If you have been listening along with us, visualizing this architecture, we hope you walk away with a completely transformed, deeply analytical vision of what it truly means to look through the eyepieces of a microscope. We value your time and your intellectual curiosity endlessly, and our exploration of this field is far from over. Because next time on the deep dive, we are taking these incredible micro tools and deploying them directly onto the clinical front lines. We will be transitioning into male factor infertility and lab strategies, exploring how the laboratory leverages this exact hardware against severe, complex clinical presentations. We will dissect the surgical processing of testicular tissue biopsies, detail highly specialized, high-magnification sperm selection techniques, and break down how clinical embryologists handle the microscopic realities of severe oligospermia and azoospermia. You absolutely won't want to miss that discussion. Until then, keep questioning the systems around you and keep exploring the architecture of the invisible.